Hello and welcome to our poster. My name is Mudassar Iqbal, and on behalf of my co-authors, I will present a quick introduction to our new clustering method for single-cell ATAC-seq data, which explicitly models the sequencing depth of individual cells. Single-cell and single-nucleus ATAC-seq data is now commonly used to study chromatin accessibility genome-wide, and the resulting peaks by cell matrix is typically very high dimensional, sparse, and essentially binary. As illustrated in this here for a synthetic data in 3 clusters, sequencing depth of a cell, which here amounts to number of ones, that is number of open chromatin sites, is an important sequencing artifact and explains much variance in the data. It can impact the downstream analysis, especially clustering. And typical workaround is to transform binary data and ignore the top latent component before clustering. The example here shows that how it affects flex mix, which is a standard binary mixture model, as it produces clusters defined almost exclusively by differences in the sequencing depth, as we can see in the bottom for the clustering for 2 clusters and 3 clusters. We instead develop a probabilistic modeling approach without transforming or ignoring any data, and use cell sequencing depth as a parameter of the model, as shown in the slide, where we have a Bernoulli mixture model set up, but the underlying distribution is modified with alpha n, representing n cells sequencing depth, which can be estimated from the data. Here, the introduction of this alpha complicates the expectation maximization updates, which are commonly used for model inference for standard Bernoulli mixture models. We provide here an approximate m-step update, which is efficient and works quite well in practice for many applications we have used. Using our model, we also provide a feature selection approach to select most informative peaks for clustering. But importantly, we develop a cluster splitting and merging heuristic at the bottom of the slide, which is explained. This heuristic is, we use it to automatically find the number of clusters in the data. In terms of validation, we start with a synthetic validation, and our method works very well in the synthetic validation experiment, where we have 8 clusters, and each of them have cells with very low and very high sequencing depth, as shown in the top left subplot. We can see the flex mix again doing quite poorly, while SCATAC, which is a recent popular method for single cell ATAC-seq analysis and clustering, does much better, but also it also falls for an extra cluster defined exclusively by very low read depth cells, which you can see in the these cell in the center. So application on real data. Here, these are 5,000 human PBMCs from 10x genomics validation data sets. Here also SCATAC is again finding very few low depth clusters, although our method, which is not affected by the read depth, as we can see, but it's finding comparatively much fewer number of clusters. While we are still working on extended applications of our method to other larger real data sets, we think our method is finding biologically meaningful clusters. And here we have used a smaller PBMC data again from 10x, which is 1,000 PBMCs, and we compared against clustering labels provided by 10x themselves. Our method finds 3 clusters, while 10x have 5 clusters, where one of the clusters from our method is essentially split into 3 in 10x labels. So by doing some motif analysis on using representative peaks of our cluster 3 and 3 clusters from corresponding 3 clusters from 10x, we think the underlying motifs in cluster 3 of our method and corresponding clusters from 10x are quite similar. And it might be that 10x labels here are, in this case, are all clustering the data. So that's from me, and thank you very much for listening.